Hello, today we will be doing our presentation about the cancer and the environment. And we will be talking about the chemicals and fine particles in the air. And we can understand the impact of the air pollution on cancer risk. Today we will be presenting me, Cynthia, Destiny, and Leah. Air pollution and cancer. Air pollution is one environmental factor that has been linked to an increased risk of cancer. These substances are known to cause the damage to cells in the body, mainly lung cells. This damage can lead to mutations in the DNA of cells that can cause cancer. People who live in areas with high levels of air pollution are more likely to develop cancer, as well as people who smoke are also at an increased risk of cancer caused by air pollution. Understanding the relationship between air pollution and cancer is crucial for our public health. Air pollution is a complex mixture of harmful substances that can cause cancer. The main sources of air pollution are emissions from vehicles, industrial activities, and fossil fuel combustions. This pollutant can cause damage to the cells in the body, particularly in the lungs, leading to mutations in the DNAs of cells that can cause cancer. Numerous studies have investigated the relationship between air pollution and cancer risk. A 2013 systematic review of 15 epidemiological studies found that the exposure to particulate matter, PM, increased the risk of lung cancer. Another study, published in 2016, found that the exposure to nitrogen dioxide and PM increased the risk of lung cancer. A meta-analysis of 27 studies published in 2019 found that the exposure to PM increased the risk of lung cancer with a pooled relative risk of 1.16. The World Health Organization, known as WHO, estimates that the outdoor air pollution caused approximately 1.8 million premature deaths worldwide each year, with lung cancer being one of the leading causes of death. The U.S. Environmental Protection Agency has designed particulate matter with a diameter of 2.5 micrometer or less, PM 2.5, as known as human carcinogen. Research has also shown that people who live in areas with high levels of air pollution are more likely to develop cancer. A study published in 2019 found that living in areas with high levels of PM 2.5 was associated with an increased risk of lung cancer mortality. Another study published in 2020 found that living near major highways was associated with an increased risk of lung cancer incidence. The National Cancer Institute has identified air pollution as one of the top environmental factors. Linked Agency for Research on Cancer has classified outdoor air pollution as carcinogenic to humans, indicating that it is likely to cause cancer in humans. In conclusion, the scientific evidence suggests that air pollution is a significant environmental factor linked to an increased risk of cancer, particularly lung cancer. Exposure to particulate matter and nitrogen dioxide has been consistently linked to an increased risk of lung cancer, and living in areas with high levels of air pollution is associated with an increase of risk of lung cancer incidence and mortality. Now let's talk about the fine particulate matter PM 2.5. These are tiny particles that refers to microscopic particles with a diameter of 2.5 micrometers or less. They come from various sources like vehicle exhaust where cars, trucks, buses, and motorcycles produce PM 2.5 pollutions through the burning of gasoline, diesel, and other fuels, industrial facilities where factories, power plants, and refineries release PM 2.5 as a byproduct. And wildfires because smoke of fireplaces, wood stove, and agriculture burning release PM 2.5 into the air. Wind-blown dust from dust storms and windy conditions can stir up dust particles from natural sources like soils and deserts, or dust from construction activities can be a source of contributions to PM 2.5 levels. An example of this can also include the traffic on unpaved roads that quick up dust particles or tiling particles and other agricultural activities. PM 2.5 can also travel long distances through the atmosphere, so areas with seemingly low productions could still be exposed to PM 2.5 from other regions. Inhalation of these particles can penetrate deep into the lungs and damage the lining of the airways, which often leads to an inflammations and mutations in the DNA of cells, causing cancer to develop. Increased oxidative stress can also generate free radicals contributing to the development of cancer. Studies showed an increased risk of lung bladder cancer, breast cancer, and colorectal cancer in association with PM 2.5 exposure. Talking about the polycyclic aromatic hydrocarbon, known as PAHs, are a class of chemicals characterized as organic compounds formed by the incomplete burning of coal, oil, gas, wood, tobacco, and other organic matter, which has been linked to an increased risk of cancer because they can lead to the DNA damage causing manipulation in genes that control cell growth and divisions that can lead to uncontrolled cell growth. Molecular mechanisms to PAHs are that PAHs attacks to the DNA causing mutations. PAHs support cell survival and proliferations by activating pathways such as AHR. Inflammations and ROS productions by PAHs results in more mutations and tumors promoting conditions. Because PAH obstructs DNA repair processes, mutations can accumulate and have a role in cancer development. PAHs are found in various environmental sources, including air, water, and soils. Exposure to PAHs occurs through inhalation of polluted air, ingestion because consuming grill or barbecue meat, especially shared portions, can introduce PAHs into the body. Skin contact usually leads to the long-term exposure that can, in certain occasions, to PAHs containing substances like coal, tar, or asphalt. Cancer most associated with PAH exposures include lung, skin, bladder, and gastrointestinal cancers. The risk of cancer from PAH depends on the type of the amounts of PAH in a person is exposed to, as well as their individual susceptibility.
Benzene is a well-established human carcinogen. It increases the risk of leukemia, particularly acute myeloid leukemia, or AML. The leukemia risk comes from long-term exposure to high levels of benzene in the air, which has been linked to an increased risk of acute myeloid leukemia, which is a type of blood cancer. Blood cancers like acute lymphocytic leukemia, chronic lymphocytic leukemia, and some non-blood cancers like multiple myeloma and non-Hodgkin lymphoma. Exposure to benzene can occur in various ways, some of which include occupational exposure. Workers in certain industries, such as those involved in petroleum refining, chemical manufacturing, or gas stations, may be exposed to benzene through inhalation. Environmental exposures can be present in low levels in outdoor air from vehicle exhaust and industrial emissions. Contaminated water is another possible source, although it's uncommon. Drinking water can be contaminated with benzene from industrial waste or leaking underground storage tanks. Consumer products, like some glues and dyes, may contain small amounts of benzene. The Environmental Protection Agency have set regulations to limit benzene exposure in the air, water, and in consumer products. Formaldehyde is classified as a known human carcinogen by the National Toxicology Program and a carcinogen to humans by the International Agency for Research on Cancer, which is based on evidence from occupational exposure. Formaldehyde exposure has been linked to certain types of cancer in humans, although the exact risks are still being studied. Sources include occupational exposure from manufacturing, construction, chemical, and production. It is linked to nasopharyngeal cancers, which is the cancer of the upper part of the throat behind the nose, sinus cancers, which develop in the paranasal sinuses located around the nose, and leukemia, particularly myeloid leukemia, which is a type of blood cancer. Exposure concerns come from people who work in industries that use formaldehyde, such as funeral homes and embalming, wood product manufacturing, construction, chemical manufacturing, textile production, and formaldehyde resin production. Arsenic is a chemical element that increases cancer risks as well. Chronic exposure is a major risk for skin cancer, bladder cancer, and lung. Industrial processes and contaminated water in certain food is usually the source for arsenic, and it disrupts DNA repair mechanisms, which causes chromosomal abnormalities, which alters cellular signaling. Molecular mechanisms. For fine particulate matter, or PM2.5, it activates pro-inflammatory pathways, which induces oxidative stress, leading to DNA damage and mutations. Polycyclic aromatic hydrocarbons, or PHAs, bind to DNA, causing mutations. They activate pathways involved in cell proliferation and survival. Benzene damages bone marrow cells, leading to chromosomal abnormalities and interferes with DNA repair mechanisms. Formaldehyde, the exact mechanisms are still in investigation, but it induces DNA damage and alters cell signaling pathways. Arsenic disrupts DNA repair mechanisms and alters cell signaling pathways. Pathogenesis. DNA damage, fine particulate matter, and some chemicals in air pollution generate free radicals, which are highly reactive molecules that damage DNA. This damage can lead to mutations in genes that control cell growth and division, potentially initiating cancer. Chronic inflammation from air pollution can cause chronic inflammation in the lungs and other tissues. This inflammatory environment can promote cell proliferation and create a favorable microenvironment for tumors to develop. Disruption of cell signaling comes from some air pollutants, which can interfere with cell signaling pathways, leading to uncontrolled cell growth and survival. Targeted therapies for cancers. Lung cancer. Targeted therapies exist for lung cancers, which is driven by specific mutations in genes like EGFR and ALK. Bladder cancer, certain targeted therapies target abnormalities in proteins on pathways involved in bladder cancer development. Skin cancer, some skin cancers like melanoma have targeted therapies that block specific proteins that are crucial for their growth and survival. Identifying the target, researchers identify specific molecules or pathways involved in cancer cell growth and survival. This leads to the development of a targeted drug. Drugs are designed to target these specific molecules, disrupting their function and hindering cancer cell growth. This leads to types of personalized treatment, which ideally targeted therapies can be tailored to the specific mutations present in the patient's concerns, leading to more precise and effective treatment. Matching therapy to exposure, unfortunately, directly linking air pollutant exposure to specific mutations in a patient's cancer is complex. Combination therapy, where targeted therapies are often used in combination with other treatment modalities like surgery, chemotherapy, or radiation for optimal outcomes. Continuous research, identifying new targets, and developing even more effective targeted therapies remains an active area of cancer research. Overall, while air pollution increases the risk of certain cancers, targeted therapies offer promising avenues for treatment based on the underlying mutations driving the cancer, regardless of the initial cause.
Detailed statistics and distributions, the global impact. The World Health Organization estimates that air pollution contributes to over 2 million deaths from lung cancer annually, with a significant burden on low and middle income countries. Variations by pollutant. The specific risk and distribution of air pollution. Related cancers vary depending on the predominant pollutants in a region. For instance, areas with high levels of industrial activity might see higher rates of lung cancer associated with PAHS. Socioeconomic factors. Socioeconomic factors often play a role. Low-income communities may have higher exposure to air pollution due to proximity to industrial sites or power plants, further increasing their cancer risk. Epigenetic alterations. Air pollution exposure may lead to epigenetic changes, which are modifications to gene expression that don't involve changes in the DNA sequence itself. These can involve DNA methylation patterns and histone modifications, potentially silencing tumor suppressor genes or activating oncogenes. Inflammatory mediators. Air pollution exposure can trigger the release of inflammatory molecules like cytokines or chemokines. Chronic inflammation creates a tumor-promoting microenvironment that fosters cell proliferation and survival. NF-κB pathway. The activation of the NF-κB signaling pathway promotes inflammation, cell growth, and inhibits apoptosis, which is programmed cell death. MAP kinase pathways. Air pollutants can activate MAP kinase pathways like ERK and JNK, which can contribute to uncontrolled cell growth and survival. PAHS. These can directly bind to DNA, forming bulky adducts that disrupt normal DNA replication and transcription. They can also activate pathways like aerohydrocarbon receptor signaling, which can influence cell proliferation and differentiation. Benzene. This is a chemical that can change bone marrow and disrupt the mitotic spindle during cell division, leading to chromosomal abnormalities. It may interfere with DNA methylation patterns, which impacts gene expression. Arsenic. Arsenic disrupts various cellular processes, including DNA repair and cell signaling. It can also alter methylation patterns and promote genomic instability. The microbiome connection. Emerging research suggests a potential link between air pollution, the gut microbiome, and cancer risks. Air pollutants might alter the gut microbial composition, which could influence immune function and indirectly promote cancer development. For our research findings, we chose an article called Air Pollution and Cancer, Biomarker Studies in Human Populations. Prospective studies indicate that air pollution likely raises lung cancer risk. Key U.S. studies, such as the Adventist Health Study on Smog, the Harvard Six Cities Study, and the American Cancer Society Study, all report higher lung cancer risk linked to urban air pollutants like PM10, which is particulate matter inhalable particles, and PM 2.5, which is fine particles. Biomarkers can be useful to understand the mechanisms and to characterize high-risk groups. Here, biomarkers of exposure in particular DNA adducts, which are DNA that's bound to cancer-causing chemicals. They are shown as well as markers of early damage, including mutagenicity, other endpoints of genotoxicity, and molecular biomarkers of cancer. Several studies have found an association between external measures of exposure to air pollution, increased levels of DNA adducts with apparent leveling off of the dose-response relationship. In fact, as Table 1 suggests, studies in different countries have shown that levels of white blood cell DNA adducts were higher among subjects more heavily exposed to air pollutants. This observation has been made in different population categories, such as among police officers in Italy and Thailand, in residents in highly industrialized areas in Poland, and among bus drivers in Denmark. In all of these cases, the more exposed the subjects had significant differences from those who were less exposed. Biomarker studies on mutagenicity and cytogenetic efforts in humans. The HPRT mutation frequencies and aromatic DNA adducts were found to be correlated in cord blood samples from newborns of mothers living in polluted areas in Poland, thus suggesting that DNA damage in utero. Experimental work had repeatedly shown lung toxicity, inflammation effects, genotoxicity, and rodent carcinogenicity of various types of particulate air pollution. The experimental work, combined with the data on frequent oxidative DNA damage in lymphocytes in people exposed to urban air pollution, suggests that 8-dehydrodeoxyguanosine, as one of the most important promptogenic lesions on lung cancer, develops through a series of progressive pathological changes occurring in the respiratory epithelium. Molecular alterations, such as the loss of heterozygosity, gene mutations, and aberrant gene promoter methylation, have emerged as potentially promising molecular biomarkers of lung carcinogenesis. So in conclusion, air pollution increases the cancer risk, specific pollutants and their impacts, molecular mechanisms and targeted therapies, foreign particulate matter, PAHS, and benzene, formaldehyde, and arsenic are key contaminants. Air pollution and significant risk factors for various cancers, and understanding molecular mechanisms and mitigating exposure can help reduce cancer risk. And we also highlighted the need for ongoing research within this area. And here's our references.